Hello students, today we are going to start the next biomolecule and this biomolecule is lipid. So what is the lipid? The lipid is a compound we generally call is as fat or oil. Now this lipid is also made up of the same components as carbohydrate, that is carbon, hydrogen and oxygen. But if you see the carbohydrate, they have the same ratio of carbon and oxygen, where in lipids, the carbon and hydrogen concentration is very high as compared to oxygen. Oxygen is found in very less amount. So this will be called as the lipid. Next thing, we need to know that lipid is a biomicromolecule. Okay, so biomicromolecule means it is going to have the molecular weight of less than 800 Dalton. So normally what should happen that if a biomolecule have the molecular weight less than 800 Dalton, then it should be soluble in the acid. We have seen in the acid, trichloroacetic acid, in the acid test, this biomicromolecule lipid should be dissolved. But unlike the other biomicromolecules, this lipid actually do not get soluble in the acid. So it is found in the acid insoluble pool. So this is another very important point. Okay, next one, is it soluble in the water? Does oil soluble in the water? No. So it is insoluble in water but soluble in the organic solvents like ether or benzene. Right? So this is another very important point. Okay, so we don't need to know much about a lipid, but yes, the classification of lipid is important. So if you see the types of lipids, how many types of lipids we can see? We can see three types of lipids. One is the true or simple lipid. Next one, we can have conjugated lipid. Then the last type of lipid is the derived lipid. So in this true or simple type of lipid, we have the cutin, suberin, wax, etc. Next one, if it is the conjugated lipid, we can have chromolipid, lipoprotein, glycolipid, etc. Then in the derived lipid, we have some lipid-like compounds, but it is not showing the same chemical nature or chemical properties or chemical structure of lipid-like. So it is going to have some ring-like structure. So it is, as it is more like lipid, so that's why it is coming under the derived lipid. So we have some examples like terpenoids, steroids, cholesterol. These are some examples of derived lipids. So just see in this classification what we have seen, that three types of lipids we can get. One is true lipid, second one is the conjugated lipid, and the third type of lipid is called as a derived lipid. Under this, in this true lipid, what we can see that if it is in case of lipid, they are going to be made up of two components. One is called as the glycerol, another is the fatty acid. So these true lipids are found is actually formed by the bond, that is ester bond, formed between glycerol and the fatty acid. Next thing, if it is the conjugated lipid, you need to know that the conjugated lipid is formed by a lipid component. Along with that, we will have some non-lipid components. Non-lipid components, like if it is in the chromolipid, we have some pigments added to the lipid. If it is lipoprotein, then along with the lipid we have a protein component. If it is glycolipid, along with lipid, glycogen or some another carbohydrate will be there. So this will be called as the conjugated lipid. And as already I have told you about the derived lipid. Derived lipid are those lipids which are more or like the structure properties. They are showing some properties of lipids, but it is not not the esters of fatty acid and alcohol, but yes, it is some ring-like structured component. So yes, these are the different types of lipids. Now after that, let's just see if it is the normal lipid, how it is formed. Okay, so suppose I need to tell you how the simple lipids are formed. So the lipids will be made up of, the lipids are made up of fatty acid and glycerol. So it is going to have one alcohol, that is glycerol, and fatty acid. Okay, glycerol and fatty acid that will be attached to each other by a bond called ester bond. So glycerol, which is also called as the trihydroxypropane, and this fatty acids, that fatty acids can be the long chain of hydrocarbon. Okay, now just see the glycerol. The structure of glycerol will be this, and in each carbon one hydroxyl group will be added. Now if it is a fatty acid, if it is a long chain of fatty acid, suppose this is the fatty acid, one side it is going to have carboxylic acid group, and it will be a suppose a long chain of any fatty acid, suppose. Okay, then in this, what type of bond we will see? Suppose this is the ester bond forms, right? So this will be removed as water. Okay, so it will be removed, and we are going to have a bond this will be the fatty acid attached to this glycerol by this bond, and this bond will be called as the ester bond. Okay, so here this structure, this is one example of, or it is the chemical formula of the monoglyceride. If that means with the glycerol there is only one fatty acid is attached. If the second fatty acid is attached, suppose this one, then this will be called as diglyceride, and if the third will be attached, that will be called as a triglyceride. So here if one only fatty acid is attached, that will be called as the monoglyceride. Okay, next one, if suppose this glycerol, monoglyceride means with a glycerol we will have only one fatty acid. If this is a diglyceride, then with the glycerol two fatty acid molecule will be attached by ester bond. And if it is a triglyceride, then in the triglyceride we will have a glycerol and three fatty acid. So this will be the monoglyceride, diglyceride and triglyceride. Next one, if all of these fatty acids are only one type of fatty acid, then it will be called as a true fat. Okay, and if it is the different type of fatty acids are attached, then it will be the another you can have waxes. So different type of fatty acids can be added. So after that, let us move to the next part, that will be how many types of fatty acids we can get. After that, let us see the different type of fatty acids. I have already drawn the structures. It's not like that you have to remember all. So what is it? The types of fatty acids. Yes, this is given in your NCERT. So whatever the diagrams will be given in the NCERT, those structures, chemical structures, you have to see. So first here we have the two type of fatty acids. One is saturated fatty acids and second one is called as unsaturated fatty acid. So what are the differences between the fatty acids? They are saturated and unsaturated. So I have given two examples. One is palmitic acid, another is the stearic acid. So if it is palmitic acid and stearic acid, you have seen the structure. It's a long chain of hydrocarbon here also, here also. And in this unsaturated fatty acid, you also have seen two examples. One is oleic acid, another is linoleic acid. But yes, we have more than this also, linolenic acid, arachidonic acid. These acids are present. But the main differences between the saturated and unsaturated fatty acid is saturated fatty acid is going to have only single chain. Okay, only single bond is found. But if it is in case of unsaturated fatty acid, it is going to have more than one or more than one double bond. So if you see this saturated and unsaturated fatty acid, which one will be better for you? What type of food is more it's good for you? Okay, so just see. First of all, yes, in this unsaturated fatty acid also we have two types of unsaturated fatty acid, which is generally given in the, or it is written in nowadays in the different food packages. One is given MUFA and second one is called as PUFA. MUFA and PUFA. MUFA stands for monounsaturated fatty acid and PUFA stands for polyunsaturated fatty acid. So in this MUFA is going to have only one double bond. Simply I have written like this. And if it is in case of PUFA, there it's going to have more than one. Okay, more double bond. Okay, so what will happen if you see the monounsaturated, polyunsaturated and the saturated fatty acid? What is the comparison? Suppose the oil packets are given to you and one oil packet or one fat is having PUFA and another is MUFA and another is the saturated fatty acid. Which will be better for you? That will be PUFA. So the reason is in this PUFA we have more double bonds, okay, and our body can store the food in its stable form. Okay, so double bonds are not a stabilized structure. So first of all, our body will try to break this double bond and it will be converted into saturated fatty acid. Whenever we eat the unsaturated fatty acid, it will be first of all converted into saturated fatty acid and then it will be stored. So what happens, saturated fatty acid, if you take, okay, in your diet, in your food, if you take the saturated fatty acid, it will directly get stored because we don't have to convert it into any type of stable form. It is already stabilized, so that's why we can store it directly. So whenever we take the food that contains saturated fatty acid, it is readily stored in our body. That gives us the fat. And in case of unsaturated fatty acid, what happens? If it is polyunsaturated fatty acid, then our body requires more time to convert into a saturated fatty acid. And if it is MUFA, then only one double bond is present. So if you break only one double bond, it will be converted into a saturated fatty acid. So that's why our body requires very less time to convert MUFA into saturated fatty acid. So that's why in our body this MUFA can be easily stored in our body as compared to PUFA. Okay, and maybe whenever the polyunsaturated fatty acid been broken down in that period of time, maybe possible our body can utilize in another metabolic other functions, right? So that's why what we can say that PUFA is better for us as compared to MUFA, and MUFA is compared to the saturated fatty acid is good. Okay, for our health, we need to have PUFA. We should consider, we should choose PUFA over MUFA, or the saturated fatty acid. Yes, we have another example why it's not needed, but yes, we have some type of food that is called as omega-3 and another is called as omega-6. The food contain omega-3 is considered better for us as compared to omega-6. Suppose we have one chain, fatty acid chain, and here we have, suppose, carboxylic acid. In this we have, suppose, the double bonds here, okay, and here omega means this is the first carbon, this is second, this is third, this is fourth, this is fifth. This way the carbon numbers will go. But if it is omega, just remember omega 3 means you have to go in reverse direction. That means this will be alpha, beta, then suppose omega. Okay, so it will be in reverse direction if you go like this. Suppose it is omega. Then what happen in this omega third carbon, if you get a double bond, then it will be omega 3. Okay, this way in this omega-6 also on the sixth carbon, whenever you go in the reverse direction, then you will get the double bond on the sixth carbon, then it will be called as omega-6 food. So if you see why it is not needed in our body, omega-3, the food that contain omega-3 is better for us, and the food that contain the omega-6 is not good for us. So let's just move to the next part. So what we have seen, the true fat. For the first, in the classification, what we have seen, the true fat. Under the true fat, we have seen the simple lipid. We have already studied the alcohol, that is glycerol, and the fatty acid that can either be monoglyceride, diglyceride, or triglyceride. The ester bond formation I have told, that are all the simple lipids. The second one is the wax. Okay, so wax, for the wax you have to remember these are also the esters of, fatty acid and alcohol, but this alcohol should not be glycerol. So the esters of fatty acid and alcohol other than glycerol will be called as the waxes. Yes, number of animals produce waxes. In case of human being also, our ear wax is a type of wax, which is a type of lipid. So on that, gland is present in our ear, that is in the external auditory meatus, that is called as the ceruminous gland, and that ceruminous gland produces the ear wax, which is also called as cerumen. Similarly, the bee wax, the workers, they produce the bee wax. The bee wax is actually a type of palmitic acid. From the palmitic acid you form the bee wax. So I have told here bee wax is made up of the fatty acid, that is the palmitic acid. Now next one is the conjugated lipid. What I have told about the conjugated lipids, that conjugated lipids are the lipids with the non-lipid components. So there is one example given in your NCERT book, that is the structure, molecular structure of lecithin. So this is going to be very much important. If you remember, this is going to be very much good for you. The lecithin is structure is very much important. So in this lecithin, this is the structure. So it is going to have a polar head group which is soluble in water, and a non-polar region, the end, which is not soluble in water. So here this part is the polar group and this is the non-polar group. In this glycerol we have the fatty acids chain, but with this glycerol we have the choline phosphate also. So that's why it will be an example of phospholipid. In the question it gave a number of time that one example of fatty acid, one example of phospholipid is lecithin, or lecithin is a type of phospholipid which is a conjugated lipid. So the structure you have to see. Similarly, we have another type of conjugated lipids also, like glycolipid, lipoprotein, chromolipids, etc. So what are the glycolipids? Glycolipids is where with the lipid we have the glycogen, and this is present in our nervous system. The myelin sheet of the neurons are made up of this glycolipid. Next one is the lipoprotein, which is very much common. This is found in the cell membranes. Our plasma membrane is also made up of lipoprotein. Next one is the chromolipids. These chromolipids are found actually in some pigments. So what are they? They are the carotene in the vitamin A. It is going to be present. So these are the chromolipid. After that, let's just move to the derived lipids. The third type, the last type of lipid is called as the derived lipids. So just see, the derived lipids do not contain, just remember, they do not contain fatty acids. Okay, so if it is not having fatty acid in it, then it should not be a lipid. Yes, it is not a lipid, but yes, it is going to show some characters of lipids. Okay, so it shows some similar properties of lipids, so that's why it is grouped under the lipids, but otherwise it is actually the ring compounds. So different rings are there. So here the rings will be there. With this there will be a long carbohydrate, a long chain of hydrocarbon will be there. So here one example of derived lipid is steroid, one example is cholesterol. The cholesterol is most abundantly found in human body, also in animal body. We have the cholesterol, which is the precursor of number of hormones. So this is the cholesterol from where we can produce number of steroid hormones. Some of the steroid hormones are the sex hormones like estrogens. Estrogens are formed from cholesterol. Then testosterone, the male hormone testosterone, is also formed from the cholesterol. Then the pregnancy maintaining hormone, that is called as progesterone, that is also formed from the cholesterol. So that's the cholesterol, which is a kind of derived lipids. So these are called as the derived third classification. So that's all about the types of lipid you need to know. That's all about the lipids. Up to this, I hope you understand. Thank you.